I leave the desk to the first speaker of today, which is Karolis Norvaisa of the Trinity College of Dublin, which will be present non-planar porphyrins as a sensor. Okay, Karolis, the desk is yours. You have 25 minutes more or less for the presentation.
Thank you so much. I'll just share my screen. I hope you can see it.
Yes. Okay.
Excellent. So, hello everyone. Good morning. Today, my name is Karolis Norvaisa, and I'm from Trinity College Dublin, a representative of Professor Dr. Matthias Osenge, and I'm a PhD student who is going to present his research on the topic of non-planar porphyrins as sensors. So porphyrins. What are porphyrins? So porphyrins are a unique class of intensely colored pigments which are naturally found in living organisms, where they play a very important role in metabolism. So some of the very best known examples are heme, the red blood cell pigment responsible for oxygen transportation, and chlorophyll, the green plant pigment responsible for light harvesting and photosynthesis. So the strong photometric responses play an important role in chemical sensing. So in the literature, there are mainly two types of porphyrin-based sensors. So the first one, where porphyrin serves as a reporter unit, indicating any changes to the recognition unit, while in type 2, the porphyrin can serve as a recognition and a reported unit simultaneously. So, but the diversity of the porphyrin function is mainly due to the variety of the metal ions found in the core of the ring system. So here comes my goal of my research, is to explore the overlooked characteristics and applications of the metal-free porphyrins for potential analyte binding and coordination. So to achieve this goal, we have employed molecular strategies to overcome this shielding effect of the ring current to expose the inner pyrrolic units. To do so, we used heavy beta substitution that leads to a severe macrocycle deformation that yields this saddle-shaped porphyrin with exposed inner core system for potential hydrogen bonding events. So this, with some additional coordinating groups close to the active center, could lead to selective detection of some particular analytes. So the initial strategy involved protonation by non-coordinative or weak acids that would activate the sensor. And then by introducing some particular analytes, the binding agent, the porphyrin, would encapsulate the analyte, just like a lock and key principle, to produce some photometric changes. So starting with the synthesis itself, Lindsay condensation with the corresponding aldehyde and the pyrrole yielded this nitroporphyrin that was subsequently reduced to amino derivative. And then at this point, only 1 out of 4 possible atropisomers were isolated, and most of our studies were actually carried out with alpha 4 atropisomer. Upon insertion of nickel, it increased stability, and we were able to separate all of the possible atropisomeric species in a statistical mixture. So the initial testing using UV spectroscopy showed that upon protonation, in this case activation of the porphyrin, the porphyrin interacts with the analytes with phosphonic and sulfonic motifs that showed very strong changes to the spectra. So the spectroscopic changes were also found to match the colors of the solutions as well. So in its natural and activated forms, the porphyrin stands in this brown-orange color solution, while in the presence of some complexing agent, it quickly drifts to this very bright green color, and the system is completely reversible upon neutralizing the porphyrin. So upon adding some base, the complexation collapse, and the initial porphyrin can be completely restored. So overall, the colorimetric response showed an easy and quick way to identify a plenitude of complexing agents to the system. So in order to confirm that the inner core system plays an essential role in complexation patterns, a methylated analog was synthesized with nickel inside, and this to highlight the lack of UV spectroscopic changes, and this actually pinpoints that the necessary use of a free base non-planar porphyrins as an active receptor. So now that we know that the inner core system plays an essential role in selectively coordinating analytes, we wanted to further investigate the importance of the peripheral coordinating sites. How important is the protonation of them? So first of all, a prime example of analogous system without a peripheral coordinating sites, octaethyl tetraphenyl porphyrin showed little to no response to a very well established coordinating substrates. And secondly, we performed UV titration studies by pre-mixing the receptor system with pyrophosphate, and then slowly add by slow addition of the acid from upstream spectra. We can see that the porphyrin overcomes core protonation before the formation of the corresponding complex. So from this, we can conclude that the system is only active when the inner core system and the periphery of the porphyrin is protonated. So we also investigated the receptor system responses to a variety of different acids, and two of which, benzene sulfonic acid and methane sulfonic acid, showed strong changes to this spectroscopic spectra, indicating the complexation pattern. So when a non-complexing acid is present in the solution, only the hexaprotonated porphyrin is formed. However, due to the lack of any complexing agent in the system, the spectroscopical changes are very slim. On the other hand, if we use an acid that contains a complexing motif, which in this case is sulfonic moiety, so then not only hexacationic porphyrin is formed, but also simultaneously complexation proceeds. So looking at the binding of particular substrates, we have determined by Jobs' method that that phosphates and pyrophosphates bind to the porphyrin in a 1-to-1 ratio, while two of the sulfate molecules can coordinate to the active center of the porphyrin. And the displacement study showed that phosphates has the weakest association to the receptor system, as indicated by the displacement by using sulfates, while pyrophosphates could show to have the highest binding potential, as it could displace sulfates and phosphates. So the displacement studies correlated very well with calculated association constants, and which showed exceptionally high for the pyrophosphate in chloroform. To support our research, we have obtained a plenitude of X-ray crystal structures. In its nickel form, we have isolated all of the possible ultrapoisomeric species, and in their free base and charge, species, we isolated an alpha 4 and alpha 3 beta atropisomers. So looking closer to the charged species, we did got complex forms with sulfuric acids. We also got forms with the benzene sulfonic acids. But on top of that, we also isolated this porphyrin coordinated by a weak tetrafluoroboric acid, which showed that which showed protonation of only in the core system. So this also confirmed that our predictions that the core system is prompt for protonation first before the protonation of peripheral amines. So looking closer at more detail into the crystal structures of the charged porphyrins in general, a quick search on Cambridge Crystallographic Data Center, and we saw that the vast majority of deposed protonated porphyrin structures are linear, or as I like to call them, eye-shaped complexes, where two of the counteranions are coordinated symmetrically on two opposing sides. So prior to our research, there were only two known structures and known examples that showed a water molecule mediated in between the counteranions and the core of the system. So now looking at our obtained structures, we can see that sulfuric acid forms this new Y-shaped complex where two of the sulfuric acid molecules coordinate on one side of the porphyrin with the predominant number of the peripheral groups. And with the benzene sulfonic acid, it shows a very similar complexation pattern, just with additional water molecule encapsulated in between the porphyrin and the acid, forming so-called T-shaped complexation pattern. So the second part of my talk will be dedicated more to NMR analysis of the complexation, complex of and atropisomerism. So looking at the Proton NMR, the inner core system signals of the alpha 4 atropisomer, it shows two sharp singlets. So the signal coordinating above the plane of the macrocycle so shows deshielding properties moving from bisulfate to methane sulfonic acid and to benzene sulfonic acid. So by revisiting our X-ray crystal structure, we can see that the binding to the core of the porphyrin is affected by the mediated water molecule in benzene sulfonic acid. So therefore this, and as well the electronic nature of the analytes could influence this deshielding properties of the coordinated inner core protons. So the interesting part is then when a counteranion, or in this case benzene sulfonic acid, locks in the anion selective inner core system, it also reduces substantially the proton exchange rates by generating very static proton signals for higher resolution NMR analysis. So this allowed us to report an ideal case study in which all of the atropisomers can stand in a rotation restricted states. So the fingerprint-like proton and nitrogen NMR signals were found to be very specific for particular atropisomer, and which provided an important porphyrin atropisomer identification features that could be used in order to fastly identify atropisomers. So for example, the alpha 3 beta atropisomer would have 4 inner core system signals, while alpha beta atropisomer would only have 1. So the corresponding integration of the intensity of the signals to the rest of the spectra also correlates very well to the amount of protons found and visible in the inner core system region. So here is 4, and here is 4 protons distributed over 4 singlets. So this is especially important in order to analyze atropisomeric mixtures, because from the intensities and the ratios of the inner core signals, one could determine and identify the appropriate atropisomeric mixtures. So, and let's say if there is an overlap of some particular inner core signals, then one could use some different complexing agent where the signals would be located in slightly different area. However, it would still sustain the same intensities and ratios characteristic to the porphyrin atropisomers. Now, not only that the inner core system is very well resolved, but the complexation also translates to the whole receptor system, which makes an ideal NMR case study for the porphyrin atropisomers. So let's take a closer look at the most unsymmetrical porphyrin system, the alpha-3 beta. And from the first glance, you can see our previously mentioned 4 inner core system protons, as well as 8 methyl groups. That instantly shows that there has to be some sort of unsymmetry in the peripheral units itself. Also, we can see the 4 aromatic region doublets that correlates to the orthoproton signals. This shows that there are 4 different phenyl moieties in the solution. In the carbon NMR, we can clearly see all of the 60 differently shifted carbon signals. Through proton-proton-proxy analysis, we can identify the 4 sets of the aromatic region signals and 8 sets of the ethyl groups. And while Proton HSQC, just a standard 2D NMR procedure, helped us to assign the proton signals to the corresponding and appropriate carbon signals. So in order to correctly assign the aromatic region, we have performed some, firstly we performed the carbon depth 135 experiment to locate proton-free positions, while Proton HMBC was used to identify the correct sequencing of the carbons and protons in the aromatic region. So also Proton Carbon HMBC helped us to identify the corresponding mesocarbon signals. So at this point, all of the aromatic signals are identified. Now looking at the Proton Carbon HMBC of the pyrrolic units, remember I mentioned before that it is from the first glance there are 8 methyl groups, so it has to be somewhat unsymmetry, and we can clearly see here in HMBC. So the pyrrolic units, the first thing to notice is the correlation of the inner core system signals to the corresponding peripheral carbon signals, and this shows how important is very well defined inner core protons for complete characterization of atropisomers. Each of the inner core signal correlated to 2 of the alpha carbons and 2 of the beta carbons, and from there we can work our way to the corresponding ethyl groups. So at this moment we have assigned every signal at the corresponding pyrrolic and aromatic sets. However, the right sequencing on how the phenyl is connected to which of the pyrrole is still unknown. And this is where through space NMR, or in this case proton-proton Rosy, comes in very handy. The interaction between the ortho position protons to the corresponding ethyl groups shows that they has to be in close proximity, and this can tell us the exact sequencing of which of the pyrrole stands in close proximity to which of the aromatic region. So Also, the inner core system signal showed the correlation to the neighboring inner core protons, which confirmed our prediction of the right sequencing. So the fact that we can see all of the NMR signals so clear, it makes actually a perfect handle in order to investigate the chiral substrates in the future. On the technical perspective, any changes to the receptor system should be clearly visible by the NMR analysis, as the chiral information could potentially be translated to the host. So the characteristics system signals are closely related to the symmetry operations found in anthropoisomers. Therefore, upon detailed assignment of all of the protons and carbon signals of all of the anthropoisomers, the symmetry order was found twofold lower than in comparison to the suggested planar porphyrin anthropoisomers. So the basis for this decrease lies within this saddle-shaped deformation of the porphyrin, actually making a distortion a molecular engineering tool to achieve symmetry reduction in microcycles. So if one would like to reduce the symmetry for a potential chiral inducement of some particular receptor system, nonplanarity could be a key to do it. So to quickly summarize my projects, I have shown that selective coordination of analytes to the inner core system of nonplanar porphyrins with specially designed binding pockets can be observed using UV-Vis and simple colorimetric techniques, while X-ray and Proton NMR analysis provided a better understanding on the binding modes itself. Upon coordination, the well-restricted system allowed for detailed NMR analysis that showed distinct inner core system signals for quick identification of anthropoisomers, and the characteristic signals were found to be closely related to the symmetry operations found in the anthropoisomers. So now that we have a system that can selectively bind analytes, this with symmetry manipulation technique, in the future our goal is to investigate the chiral substrates and for chiral recognition. And in the bigger picture, we aim to detect and eliminate the chiral pollutants from our environment. So I would like to especially thank my supervisor, Professor Dr. Matthias Osenge, all of my dearest colleagues and technical staff, all of the funding agencies, of course, especially Initio for organizing this lovely event today. Thank you everyone for your attention.
Okay. Thanks, Karolis, for the nice presentation. We didn't have a question on the chat, so if someone wants to ask, okay, there is a question right now. I read the question, so I have a question regarding slide 4. Okay, you can read in the chat. 14. Did you try to test the behavior of chiral sulfonic acid in a racemic form when at least 2 molecules of sulfonic acid bind to porphyrin? This should result in the diastereomeric complexes, which will be distinguishable by NMR. Might be useful for enantiomeric measurements.
So, sorry, I just want to see the question. Okay, now I see it. 14. Try to test behavior. Oh, chiral sulfonic acids in racemic forms. So actually, that's our goal, right? In the future, is to investigate more of a pronounced chiral substrates. So something like camphor sulfonic acids that has 2 chirality centers, S and R, would be a perfect target in terms of analyzing the activity of the receptor systems. And this is actually our potential plans and potential future plans, is by using simple NMR techniques and using a variety of different porphyrin receptors to use chiral guests and see if the chiral information can be translated to the porphyrin itself. So by doing some carbon NMR measurements, proton NMR measurements, to detect which of the groups are being affected by the chiral guests and which of the tropizomers maybe is not. So this is obviously our very close future plans, and we will be working on this very close.
Okay. Okay. More questions? Okay, if not, we can thanks Karolis for the nice presentation, and we can move to the next speaker,